Welcome back, everyone. I hope the morning session so far has been given you plenty to think about. And we're moving now from the broad horizon of environmental monitoring through to a detailed look at what it takes to navigate the LAL transition. In this session, I'm pleased to be joined by Dr. Harman Hauer. Dr. Hauer is head of microbiology at Panpharma and also runs Hauer Consulting. And he has direct hands-on experience of making this kind of transition. He's going to walk us through the scientific landscape, practical laboratory realities, and some lessons that he's learned when moving towards non-animal derived reagents. We'll have time for questions at the end, so please do submit them on the Swapcard platform. Use the Q&A panel. We'll get through as many as possible. And Harman, I'm going to hand over to you.
Good afternoon, everyone. First of all, thank you, Ian, for the very kind introduction, and thank you all for joining this session. Today, I would like to discuss what it really takes to move beyond the Limulus imiboside lysate-based tests and successfully implement modern, sustainable endotoxin testing in a regulated GMP environment. And for me, this is highly relevant from both a quality and sustainability perspective, as basically for decades, horseshoe crabs have played a critical role in protecting the quality of parenteral drug products and therefore countless patient lives worldwide. And in that sense, they've become incredibly important in pharmaceutical quality control and should probably be considered, as depicted in this logo, as holy crabs. And in this talk, I will revisit the fundamentals of pyrogen and endotoxin testing, review available methodologies, and discuss recent developments in the field. But most importantly, I will share some of our practical experience implementing recombinant factor C-based methods. And this includes both the strategic business perspective, verification studies, and also the routine use in the lab, for which I will share some examples of practical step-by-step protocols. And in the end, I will present data from using modern RFC-based systems to discuss what these results could mean for laboratory who are considering a transition away from the traditional LAL. And hopefully by the end of the session, we can have some time for questions and a good discussion. And now I think I will turn my camera off so it doesn't overlap with the presentation slides, but I will turn it back on later for the discussion. So before we start discussing endotoxin testing itself, it is worth taking a step back and asking a very simple question. Why do we actually need to test for pyrogens in the first place? Pyrogens, by definition, are substances that are capable of inducing fever in humans or animals, and consequently, they present an unacceptable contamination risk for medicinal and drug products, medical devices, and any product really intended for patient administration. From a pharmaceutical perspective, pyrogen control is therefore a critical responsibility, not only for manufacturers, but also regulators, healthcare professionals, and ultimately for patient safety. Now, pyrogens are actually a broad group of substances. They can originate from bacteria, fungi, yeast, viruses, plant-derived materials, small molecules, or even process-related impurities. In other words, many different contaminants are capable of triggering pyrogenic responses. However, when discussing pyrogen testing, we most often focus on one specific subgroup, and that's the bacterial endotoxins, which are lipopolysaccharides, or in short LPS, located in the outer membrane of gram-negative bacteria. Structurally, they consist of three major regions: lipid A, which is primarily responsible for the biological activity, then a core oligosaccharide, and the O antigen, which is the primary determinant of antigenic specificity, which is shown also in the figure on the left. This illustrates the general structure quite nicely. And although endotoxins can differ substantially between bacterial species, and even small structural differences may heavily influence their biological activity or detectability, in fact, they all share one important characteristic, and that's they trigger a powerful innate immune response. And as such, if introduced into patients at sufficient levels, endotoxins can induce inflammation, fever, endotoxemia, sepsis, septic shock, multiple organ dysfunction, and death of the patient. And this remains highly relevant today, as despite the tremendous advances in modern medicine, sepsis continues to be associated with substantial mortality rates worldwide. And depending on patient pool, region, or study design, reported mortality rates for severe sepsis and septic shock frequently remain in the range of approximately 20% to 50%, which is absolutely crazy if you ask me. So for that reason, endotoxins are generally regarded to as the most critical pyrogens in terms of pathogenicity and contamination risk in the pharmaceutical manufacturing industry. They are widespread in the environment, comparatively easy to introduce into manufacturing processes, for example, by systematic contamination with gram-negative bacteria, and they are associated with significant clinical risk. All of this combination of high abundance and high pathogenic potential is exactly why pharmacopoeias around the world place such a strong emphasis on pyrogen and endotoxin testing as an essential component of pharmaceutical quality control. So where do endotoxins and gram-negative bacteria actually come from? One of the most important sources is obviously water, and any communal and industrial water system and process that requires water. We recently conducted a small study on the microbiome of our local drinking water system, and in line with numerous publications in this field, we found that approximately 85% of the identified species were gram-negative bacteria. And since these organisms naturally carry endotoxins within their outer membrane, every water system contains a very individual endotoxin burden. In fact, it is really well established that drinking water systems commonly contain detectable endotoxin levels that vary substantially and can commonly be found in the range of 5 to 100 endotoxin units per ml, with critical peaks reaching way higher than that. And for comparison, pharmacopoeia limits for water for injection would be 0.25 endotoxin units per ml. Now, of course, every water system is unique and species composition varies depending on location, season, materials of the construction, biofilm release events, and many other factors. And the key message here is that drinking water systems basically represent one of the largest reservoirs of endotoxin-producing bacteria, and therefore are one of the most relevant upstream contamination sources for pharmaceutical manufacturing. Now, for the general population, drinking water is typically considered safe, and endotoxin exposure through routine consumption is usually not regarded as a significant health concern. However, the situation changes completely when we look at pharmaceutical manufacturing, because virtually every pharmaceutical facility relies on water in multiple processes. Whether you manufacture APIs or drug product, medicinal devices, or perform laboratory testing, water is involved everywhere. And while it undergoes purification steps such as reverse osmosis, deionization, filtration, distillation, the incoming water remains the starting point of this entire process. And the water is only one part of this picture, because facilities, utility systems, raw materials, packaging components, as you can see here on the right, equipments, room environments, and personnel all can contribute to endotoxin contamination if they're not adequately controlled. And that is why endotoxin control should always be considered as part of a comprehensive contamination control strategy for any pharmaceutical production site, supported by a risk-based hygiene concept, routine monitoring, trending, and where appropriate, the deeper understanding of the microbial population present within this facility. One final point to this slide: steam sterilization may reduce endotoxin levels, but it does not reliably eliminate them, and therefore autoclaving cannot be considered an effective depyrogenation strategy. So when it comes to endotoxin control, the most effective approach is still prevention, specifically through robust water systems and generally well-constructed manufacturing environments. So let's check out some milestones of endotoxin testing methodologies. We just established basically that pyrogen and endotoxin testing is essential. So pyrogen testing first became mandatory for injectable products in the 1950s. And it was initially performed using the rabbit pyrogen test, or RPT. And as most of you know, this method relies on monitoring the temperature response of rabbits following the administration of the test solution. A major breakthrough, or as I like to call it, the first endotoxin testing revolution, then occurred in the late 1960s and early 1970s with the discovery that horseshoe crab blood contains an enzymatic cascade that reacts extremely sensitive to the presence of bacterial endotoxins by clotting the blood. And this led to the development of the Limulus ameboside lysate test, or LAL test, which became the foundation of modern endotoxin testing. And depending on the horseshoe crab species used for the test, the lysate was referred to as, for example, LAL or TAL, but the underlying principle remains the same. So the original gel clot method used, which we will look at in just a second in detail, which today corresponds to method A and B in the European Pharmacopoeia, provided a significant improvement over rabbit pathogen testing, but still required a relatively high work effort, which in turn means they are difficult to scale. Later, as an analytical technology, advanced automated reader systems using kinetic turbodimetric or chromogenic detection principles were introduced, and these methods, representing method C through F in the European Pharmacopoeia, substantially improved throughput, standardization, data integrity, and today they represent the predominant approach in most laboratories. But despite these technological improvements, all of these methods still rely on lysate from horseshoe crabs. So to reduce this dependency, recombinant alternatives were developed, and the first recombinant factor C assay became available already in 2003, and providing a method that retained the biological specificity for endotoxin detection while eliminating the need for animal-derived lysates. Regulatory acceptance, as it is within the pharmaceutical industry, however, that took time. And you can see here in 2012, regulatory authorities clarified that recombinant methods could be used provided manufacturers demonstrated comparability and compliance with existing endotoxin testing requirements, which basically meant a full method validation. This was followed by some other important milestones, including the introduction of the monocyte activation test, which was actually 2 years before that. And the MAT is particularly interesting to me because it's based on human immune cells, and therefore it allows the detection of a much broader range of pyrogens. And then, of course, the dedicated RFC chapter came later in 2021, 2.632 in the European Pharmacopoeia, which is soon to be included also in 2.614, and of course publishing of the USP 86 in 2023. Finally, in 2024, another major transition started. This is the discontinuation of the rabbit pyrogen test under European Pharmacopoeia, which basically was a decision to remove the remaining rabbit pyrogen testing texts with a completion goal by 2026, but this was mostly already implemented in 2025. Overall, the trend is quite clear. Over the last 70 years, pyrogen testing was continuously evolved towards methods that are more sensitive, more standardized, more sustainable, leading to the current development towards recombinant methods in MAT, in my eyes presenting the second big endotoxin testing revolution after the discovery of LAL. Now let's dive into these different methods real quick, and let us start with the rabbit pyrogen test, or RPT, which for many decades represented the standard approach for pyrogen testing and pharmaceutical quality control. And the principle is relatively straightforward. The test substance is injected into a group of rabbits, and their body temperature is monitored over a defined period of time. And depending on the cumulative temperature increase observed across these animals, the sample is classified as either a passing or failure of the test. One important characteristic of the rabbit pyrogen test is also that it measures a physiological response in a living organism rather than detecting a specific reaction. And as a result, the method is capable of detecting a broad range of pyrogenic substances, of course, and not only endotoxins. At the same time, this broad biological response also comes with limitations. The method provides only limited opportunities for quantification. It does not offer many of the analytical controls that we routinely expect from modern assays, such as standard curves, spike recoveries, or qualitative positive controls. And in addition, results may be influenced by a variety of factors, such as animal variability, environmental conditions, and differences between laboratories. So from an operational perspective, the assay is comparatively resource intensive. It requires dedicated animal facilities, extensive preparation, specialized personnel, and considerable testing time, all while still giving very minimal sensitivity. However, by far the most important discussion surrounding this test is no longer analytical performance, but definitely animal welfare. So over the last decade, regulatory authorities, pharmacopoeias, industry stakeholders have increasingly promoted the replacement of animal-based testing wherever scientifically appropriate alternatives are available. And this is the case for RPT, and this development has facilitated the removal of pharmacopoeia RPT tests in Europe. So in many ways, the rapid pyrogen test therefore presents the transition point between the historical foundations of pyrogen testing in general and the modern generation of cell and lysate-based and recombinant technologies. So as mentioned earlier, the introduction of the limulus ameboside lysate, or LAL assay, marked the first major revolution in endotoxin testing. Both LAL and TAL are based on the same biological principle. They utilize lysates obtained from the blood of horseshoe crabs and exploit a natural occurring defense mechanism in their immune system against gram-negative bacteria. In a simplified view, endotoxins activate the protein's factor C, which initiates an enzymatic cascade involving factor B and the clotting enzyme, which ultimately leads to the formation of coagulin and the visible clotting of the reaction mixture. And the practical execution is actually quite simple. The lysate is mixed with this test solution and incubated under controlled conditions, and if sufficient endotoxin is present, then a stable clotted gel forms. If no clot is formed, then the sample is considered negative at the tested sensitivity, which sometimes is tough to see as there can be like intermediate states. But compared to the rapid pyrogen test, the gel clot method offers several important advantages. Most importantly, it is highly specific for endotoxins, provides defined positive controls using standardized reference endotoxins. It requires no animal testing during routine execution of the test, and it is considerably easier to standardize between laboratories. However, this specificity also represents one of the limitations, of course, as the test does not detect the broad range of pyrogens as the RPT does. In fact, it is designed specifically for bacterial endotoxins and therefore cannot identify non-endotoxin pyrogens. Another well-known limitation is the potential interference caused by certain glucans. These compounds can activate factor G, a separate component of the horseshoe crab clotting cascade, and may therefore produce like a false positive reaction if not adequately controlled. So despite these limitations, the gel clot method established the foundation of modern endotoxin testing, and it remains an accepted pharmacopoeial method worldwide. Many of the more automated endotoxin assays that we use today are ultimately built upon the same biological principle. And the next major step in the evolution of endotoxin testing was not a change in biology, but rather a significant improvement in analytical detection. Both kinetic, turbidimetric, and chromogenic assays are still based on this horseshoe crab clotting cascade that we discussed on the previous slide. Endotoxins activate factor C, which initiates the enzymatic reaction, ultimately leading to activation of the clotting enzyme. The difference lies in how this reaction is measured. In kinetic turbidimetric assays, the increase in turbidity caused by clotting is monitored automatically using optical measurements. In other words, instead of visually assessing whether a gel clot has formed, the instrument continuously measures changes in light transmission and its absorbance, as depicted here. In chromogenic assays, the activated clotting enzyme cleaves an additional synthetic chromogenic substrate, releasing a colored compound that can be measured photometrically. The stronger the color development, the higher the endotoxin concentration. And the major advantage of both kinetic and chromogenic approaches is automation and quantification. Unlike traditional gel plot methods, these systems provide quantitative results, standard curves, objective endpoint determination, positive controls, and routine recovery testing. This greatly improves the reproducibility, data integrity, and suitability for high sample numbers in controlled GMP laboratory environments. So as a result, kinetic turbidimetric and chromogenic methods have become the dominant endotoxin testing technologies in many pharmaceutical laboratories, especially in Europe and North America. But they still share some of the same fundamental limitations as all lysate-based assays. Interference from certain glucans remains possible. And many of you, I'm sure, are familiar with another challenge, product-specific inhibition or enhancement of the enzymatic activities. So depending on the product matrix, endotoxin recovery can be affected and method optimization may be needed. Typical approaches include dilution strategies, buffering, sample preparation adjustments, or the use of specific demasking reagents. And in most cases, these challenges can be managed, but there are still products that remain difficult to be tested reliably using these conventional lysate-based methods. And that's normal because it should be clear that there is no one single biological assay that can be used for all sample types. Maybe 90% or 95% of products can be tested with one system, but there's not one method for everything. So one strategy to reduce the dependence on horseshoe crab lysate is the use of recombinantly produced enzymes, either in the form of recombinant factor C, shown here, or as a recombinant cascade, also called RCR, which comprises RFC, RFB, and recombinant clotting enzyme. Generally, RFC consists of an LPS binding domain and a protease domain, and upon binding endotoxin, the protease domain becomes activated and cleaves a fluorogenic substrate, allowing quantitative detection via fluorescence measurements, which you can see depicted here. And in our hands, this assay has proven to be a specific and sensitive method. In comparison, the recombinant cascade reagents works very similarly to the classical chromogenic LAL detection that we discussed earlier, except, of course, that all enzymes are produced recombinantly rather than being isolated from horseshoe crab lysate. And an important advantage, I think, of both systems is that they appear largely unaffected by the previously discussed glucan interference, as the alternative activation pathway via factor G is not present in the recombinant reagents. In terms of availability, recombinant endotoxin testing is no longer a niche technology. Today, several RFC and recombinant cascade-based systems are commercially available from European, American, and Japanese manufacturers mostly. So how to conduct RFC analysis in the lab? And in this example, we utilize Lonza reagents. First, the stock solution of the standard is prepared by dissolving the lyophilized endotoxin reference standard in a defined volume of endotoxin-free water. This is very similar to standard LAL tests, with the main difference being the concentration of the stock solution. While some LAL methods use stock solutions with, for example, 100 endotoxin units per ml, the concentration for this RFC assay is 20 endotoxin units per ml. From the stock solution, a series of dilutions with endotoxin-free water is used to prepare 4 standard solutions with concentrations ranging from 5 endotoxin units per ml to 0.005 endotoxin units per ml. So in this case, there's a higher sensitivity as in most LAL-based assays. The resulting standard curve, for which you can see an example here on the left, is also very similar to what we know from the established LAL systems, with the exception that we have an increasing fluorescence unit count with increasing standard concentration in comparison to the negative slope that we were used to as a result from the measurements over time that we see, for example, with turbidimetric assays, where detection time is lower with increasing standard concentrations. But overall, curve parameters with the correlation coefficient, slope, the y-intercept, which you can see here on the right, remain basically the same. And I believe the fact that many steps remain similar or identical is also a major advantage for transfer, as laboratory staff only needs to train very few new procedures and steps if they are already used to modern LAL testing systems. This is also the case for the CV, the coefficient of variation, which is calculated for every standard, routine sample, and positive control, as these are all performed in duplicates. So preparing samples generally follows the same procedure as with LAL tests. The samples are diluted with endotoxin-free water or the solvent in accordance with the respective method verification to minimize interfering factors, such as inhibition or enhancement. Next, 100 microliters of each standard solution and each diluted sample are pipetted into the designated wells of the 96 well plate according to the plate layout and defined in the WinKQCL software or any adjacent software that you're using. Depending on the supplier or system you are using, you may, of course, have different software, but overall, our experience was that the WinKQCL software is one of the most intuitive and practical solutions. Furthermore, I would highly recommend to design your workspace in such a way that you do not need to print out these layouts, but that you can directly copy the layout from the program, from the monitor. And it is never a good idea to see printouts with additional comments, arrows, and other changes in a GMP laboratory environment. An important aspect of working with RFC, as it is with most enzymatic assays really, is consistently maintaining the cold chain for all reagents. A break in the cold chain can lead to loss of enzyme activity, and this will show in the reproducibility of your results. The only aspect that can be tricky is that before you start your measurements, the required reagents must be allowed to reach room temperature, but should not remain at room temperature for too long afterwards. So to do this, the individual components should be removed from the refrigerator only shortly, like 15 to 20 minutes before use, and stored away from sunlight until the needed for assay preparation. And this is particularly important for the substrate since the excitation wavelength of the fluorophore is present in sunlight, and hence exposure to light can affect the measurement. So we would advise to place a lid or a turned over box over the reagents during this time. For the positive product control, or PPC, 10 microliters of the 5 endotoxin units per ml standard solution is added to the corresponding wells. In this step, you have a minimal risk of accidental carryover from these positive controls into neighboring product samples, which is why it is highly important to have qualified laboratory personnel who are trained for this method. And this is obviously the same for classical LAL tests, and laboratories who have years of experience with kinetic turbidimetric or chromogenic detection systems, they will not have an issue with this transition in our experience. But if you have never had a plate assay in your lab before, it might be smart to plan training period for your team and also to get some external help with the practical parts of implementation and training. Also, compared to some LAL methods, the PPC concentration for this RFC assay is reduced from 1 endotoxin unit per ml to 0.5 endotoxin unit per ml, which is also the result from the using different standard concentrations. I mean, you can just adjust this, but it makes sense to use a middle standard concentration for the PPC specs, right? So after adding the PPCs, the plate is transferred to the reader for pre-incubation for 10 minutes. And during these 10 minutes incubation time of the plate and the reader, the working reagent is prepared from the individual components for which the specified order should be followed, and which is first you mix the substrate and the buffer, and then you add the enzyme, and the solution is mixed gently by pipetting up and down. Strong mixing, such as vortexing, should be avoided because these forces can actually impact enzyme activity. So generally, this working reagent is prepared in a ratio of 5 to 4 to 1. That is substrate to buffer to enzyme, and the required volumes can be flexibly adjusted to the number of occupied wells / to the number of samples you want to test. After preparing this working solution, the remaining reagents should be immediately returned to cold storage, as I mentioned before. And maybe one last remark. Overall, our experience and trending shows that laboratories with standardized procedures for all laboratory steps, maybe even detailed visual step-by-step protocols like the ones here, perform way better in terms of stability between measurements, between analysts, standard curves, and CV percentages. So it makes sense to invest some time in a very clear lab protocol. Lastly, the addition of the working reagent is just like the addition of the lysate in the LAL test. It is performed after the plate has been incubated in the reader for 10 minutes using, for example, a multi-channel pipette to add 100 microliters of the working reagent into each well. And then you reinsert the plate into the reader and start the measurement. And after the measurement is completed, the software automatically evaluates all critical quality parameters before a result can be reported. First, the standard curve is assessed to confirm acceptable linearity using the correlation coefficient, the slope, the y-intercept values. Next, the precision of the standards and the templates, the samples, is verified using the coefficient of variation, so the CV, which should remain within the predefined acceptance criteria, which can be, for example, 10% or 20%. In addition, the positive product control is evaluated to confirm that the sample matrix does not interfere with endotoxin detection and that acceptable recovery is achieved. And only when all of these criteria are met can the endotoxin result be considered valid and compared against the specification limit. So generally, from a GMP perspective, this evaluation obviously is a very important step. We first verify that the entire assay performed as expected. And then if any parameter falls outside the acceptance criteria, the result must be investigated. And if necessary, the analysis has to be repeated. This evaluation process is highly comparable of what we do currently, or most laboratories do currently, with kinetic turbidimetric or chromogenic LAL systems, which also simplifies implementation and routine use of the RFC assay. So one method that we haven't touched on yet, I would really like to show you very briefly before we move on, the monocyte activation test, or MAT, which is a very interesting player in the pyrogen testing landscape. Unlike the methods we discussed previously, MAT does not rely on horseshoe crab enzymes or recombinant horseshoe crab enzymes. Instead, it utilizes human immune cells and therefore provides a highly relevant model for assessing pyrogenic reactions in humans. So in a simplified workflow that you can see here on the left, human monocytes are combined with a test substance and incubated for a defined period. If pyrogens are present, the cells become activated and release pro-inflammatory cytokines. And these cytokines are then quantified using an ELISA-based detection system, providing a robust and quantitative readout for the immune response. From an analytical perspective, MAT offers many of the advantages we already know from modern endotoxin assays. The method supports standards curve, positive controls, quantitative measurements, and parameter-based routine assay release. So what I personally find most fascinating about MAT is still that it directly models the biological responses we are ultimately trying to prevent in patients. And this figure schematically on the right shows the signaling pathways involved in the corresponding innate immune activation. For example, endotoxins activate complex intracellular cascades that ultimately lead to NF-kappa B activation and the release of endotoxin, of inflammatory cytokines. And then the cellular response can also be triggered by other pyrogenic substances, right? So this includes lipopeptides, flagellin, fungal components, and numerous additional microbial products. And that is really the key difference compared to endotoxin-specific methods. While LAL, RFC, and recombinant cascade-based assays are designed to detect bacterial endotoxins, MAT is capable of detecting a much broader spectrum of clinically relevant pyrogens. For this reason, MAT is probably one of the most physiological relevant pyrogen testing methods currently available for products intended for human use, and I'm sure we will see much more usage of MAT in the future. So now that we've looked at the individual methods, let's compare them side by side. The rapid pyrogen test, the different LAL-based methods, recombinant assays, and MAT. All have the same ultimate goal, protecting patients from pyogenic contaminants. However, they differ substantially in terms of performance, sustainability, regulatory status, and practical implementation. Starting with the usage of the classical rapid pyrogen test and the various LAL-based methods that can be used for a very broad range of sample types, including drug products, API, excipients, and water samples. But when we look at analytical performance, the picture becomes more differentiated. The rapid pyrogen test remains valuable because it can detect certain non-endotoxin pyrogens, but compared with modern analytical methods, it offers relatively low sensitivity, limited standardization, and no quantitative output. So within the LAL family, the automated chromogenic and kinetic turbidimetric systems clearly outperform the traditional gel clot method by providing quantitative results, standard curves, recovery testing, and so on. And the recombinant methods build on these advantages. And they provide highly sensitive quantitative endotoxin detection while eliminating the dependence on lysate harvested from horseshoe crabs and thus providing a much more sustainable variant. Lastly, MIT. It occupies somewhat of a unique position, as I just mentioned. While LAL and recombinant methods focus on detecting bacterial endotoxins, MAT is capable of detecting a much broader range of pyrogenic substances. And as I said before, in many ways, it is arguably represents the most physiological relevant model among the methods shown here, at least for testing drug products intended for human use. Of course, every modern method comes with its own strengths and limitations. MAT requires considerably more laboratory effort and longer assay times, as well as much higher cost per sample, while the recombinant enzymatic methods remain limited to endotoxin detection. I think, actually, method selection should always be based on the intended application, product characteristics, and overall risk assessment, so that you can get a system that is tailored to your need. One aspect that has also gained increasing importance over the last decade is supply chain security, and this is where the comparison becomes particularly interesting. While recombinant methods rely on well-established biotechnology manufacturing processes, all traditional TAL and LAL methods remain dependent on wild animal populations. And this is why I believe it is important to start planning a transfer to recombinant assay systems, if not already started, and to avoid supply chain risk, increase sustainability and business continuity, and to lower the ecological impact on horseshoe crab populations. So now I would like to make a very brief excursion to the phenomenon that has had a major impact on endotoxin testing over the last decade, and that is low endotoxin recovery, or LER. So in an aqueous solution, endotoxins do not exist as individual molecules. Instead, they naturally form larger aggregates such as micelles or vesicle-like structures. These aggregates are stabilized by cations such as calcium and magnesiums, but problems arise when formulations contain a combination of chelating agents or surfactants. Chelators such as citrate or EDTA can remove the stabilizing divalent cations and from the endotoxin aggregates, and at the same time surfactants such as polysorbates or tween can insert themselves into the weakened structures and progressively disrupt the aggregates to eventually disperse the endotoxin structures into monomeric forms. And this is where the analytical problem begins. The activation of factor C within the LAL cascade depends on the structure of the endotoxin aggregate. So monomeric endotoxin is much less efficient at activating the enzymatic cascade and may become essentially invisible from the assay. As a consequence, a sample can contain a significant amount of endotoxin while producing little or no detectable signal in the test system. What makes this particular problem very problematic, and that is also that in the standard positive product control, this can still often perform, the PPC can still often perform as expected, as the LER effect only develops over time. So in other words, the assay itself appears completely valid, while any endotoxin present in the original sample may remain undetected. So this can lead to false negative results and an underestimation of the actual endotoxin burden. So how do we verify BET analysis considering LER? And because I'm very short on time already, I think I will skip this slide and move on to the next slide, which is, do aging systems present an economic risk? Because I really want to show some of our data and also some of the business aspect before we finish the session. So far, we have mainly discussed scientific, regulatory, and sustainability aspects of endotoxin testing, but there is another perspective that is becoming increasingly important, and that's business continuity and lifecycle management. For most pharmaceutical manufacturers, bacterial endotoxin testing is a critical release test. If endotoxin testing cannot be performed, products cannot be released to the market. And that means reliability and availability of the testing system itself become business critical factors. Of course, the actual level of risk depends on the individual situation, as large organizations may operate multiple reader systems, qualify several suppliers, and maintain strong relationships with contract laboratories. Smaller organizations may have considerably less redundancy and resources available. Nevertheless, two trends can be currently observed across the industry in my eyes. First, many established reader platforms are approaching the end of their life cycle. Maintenance contracts are eventually expire, spare parts become increasingly difficult to obtain, software support may be discontinued or in need of updates, and supplier portfolios continue to evolve. For some long-term established systems, these developments are already becoming visible right now. Second, traditional endotoxin testing still relies on biological reagents that are derived from horseshoe crab populations, while strong efforts are being made to preserve these populations and stabilize supply chains. Dependence on biological raw materials inevitably introduce a level of uncertainty that recombinant technologies largely avoid. So the key question is therefore not whether a critical failure will occur tomorrow. The key question is whether an organization is prepared if it does. So if a reader system unexpectedly fails, or if a critical reagent becomes unavailable, laboratories may need to rapidly qualify replacement equipment and transfer methods to contract laboratories, or perform comparability studies, update validation or verification documentation, and potentially quickly initiate change control and regulatory activities. And even when all of these activities are executed efficiently, they require time. And time translates directly into operational risk, delayed batch release, and ultimately economic impact. So from a GMP perspective, this is fundamentally also a lifecycle management question. For that reason, I increasingly view the transition towards modern endotoxin testing technologies not only as a sustainability and animal welfare initiative, but also as an important element of business continuity and long-term operational resilience. And ideally, the transition starts while the current system is still working and not when it has already become a problem. As I mentioned before, one potential solution is to operate multiple reader systems, utilize multiple analytical methods, and maintain access to more than one supplier. In many larger laboratories, this is already common practice, but this can be highly challenging for smaller quality control laboratories and organizations with limited resources. Another interesting option then is the use of a dual reader platform that supports established LAL-based assays, for example, turbidimetric detection, and at the same time, newer fluorescence-based methods such as the RFC on the same instrument. The major advantage of such an approach is that it reduces business risk while providing a controlled and flexible transition pathway. Existing methods can continue to be used while comparability studies transfer validations, change controls, and regulatory activities are performed over time rather than under urgent circumstances. This can be particularly valuable for organizations with a broad product portfolio and numerous product-specific method verifications, because instead of transferring all methods simultaneously, verification studies can be strategically planned and implemented step by step while maintaining uninterrupted testing capabilities. There's also a scientific benefit. As we've discussed throughout this presentation, there's no perfect pyrogen and endotoxin testing method. Different products and formulations behave differently, and occasionally a test substance that performs poorly on one analytical platform may perform well on another. For that reason, maintaining access to multiple technologies or combining them within a dual reader strategy not only improves operationability, but also supply chain security, and increases methodological flexibility overall, actually. Now, in one specific example, we started to evaluate the feasibility of RFC-based endotoxin testing. And the first step was to contact potential suppliers. And I have to say that the support we received throughout the process was excellent, ranging from technical information and training to having the possibility of using demonstration systems to perform a realistic assessment in our own laboratory environment. And at the same time, we were fortunate to have really highly motivated bachelor students who carried out a comprehensive comparison between our established LAL methods and commercially available RFC systems. And believe me, this was not a trivial exercise, as we wanted to get a really good feeling for the systems, potentially by including a very broad range of pharmaceutical products in the test, and discovered in the end 75 different products and substances. To keep the assessment as realistic as possible, we deliberately avoided any method optimization during the initial screening. So first, we simply applied the same sample preparation procedures and dilution factors that had already been established within our validated LAL methods and transferred them directly to the RFC assay. And the outcome was surprisingly straightforward. Immediately, 91% of all tested products achieved a valid endotoxin recovery rate without any additional optimization. And for us, valid recovery rate did not mean just over 50%. We defined this as above 70%. So with minor changes within batches, this would still be valid. During this test, only 3 product lines showed recovery rates clearly outside the acceptable range during this initial assessment: 1 antibiotic, 1 glycosaminoglycan, and 1 vitamin preparation. And for us, these results were highly encouraging and demonstrated that the vast majority of the existing endotoxin methods could potentially be transferred to recombinant technologies with very little modification. So after the initial feasibility assessment, we wanted to challenge the methods a little further, and we investigated whether changes in the sample dilution would influence assay performance. For every product included in this initial screening study, we therefore tested both higher and lower sample dilution, in addition to the dilution already established in the verified LAL method. And again, the results were remarkably consistent. 73% of all tested products performed best using exactly the same dilution that had already been established from the LAL assay. This suggested 3 things. First, the existing LAL method verifications had already identified very suitable sample preparations and dilution factors, so good for us. Second, the transfer to an RFC-based method would likely require much less effort than we originally anticipated. And third, the RFC assay itself appeared to be a robust and stable analytical system. Interestingly, another 17% of products actually performed better at lower dilutions than those used in the established LAL methods. So from a practical laboratory perspective, this could potentially reduce sample preparation effort while maintaining optimal endotoxin recovery. And then lastly, 7% of products required higher dilutions to achieve valid recovery rates. These included the antibiotic and the vitamin preparation that I mentioned on the previous slide, but the dilution was adjusted and both could be transferred successfully. Lastly, This left only one single product line, making up 3% of the sample pool, that continued to show unsatisfactory recovery rates despite additional optimization efforts. And for this product, we evaluated alternative sample preparation, different buffers, several additional conditions, and nevertheless, the recovery for this product remained consistently low. And this observation perfectly reflects a point that I have emphasized on before. There is no single endotoxin testing method that is universally optimal for every product. Even highly robust technologies will occasionally encounter challenging formulations that require an alternative approach. But overall, the conclusion was very encouraging for us. Most drug products and raw materials could be transferred with little validation effort beyond the required comparability studies, and only a single product line proved unsuitable for the transfer within this project. So if there's one message I would like you to take away from this slide, it is depending on the product portfolio, the transition to a more sustainable and recombinant testing strategy may be considerably easier than many laboratories expect. As we come to the end of the presentation, I think one thing becomes very clear. For approximately 70 years, the rabbit pyrogen test played an important role in ensuring patient safety, and the introduction of LAL-based testing marked the first major revolution in pyrogen testing and dramatically improved analytical performance while reducing direct animal use. Today, we are standing still at the beginning of the next big transition. The technologies are available, the regulatory framework is evolving. The question is therefore no longer whether recombinant methods are technically feasible. The question is, how can we implement them in a way that is practical, sustainable, and aligned with business needs? So a few starting points may be: evaluate future investments not only from a CapEx perspective, but also from a business continuity and supply chain perspective. Consider multiple reader systems, multiple suppliers, or dual platform strategies to enable a gradual and ongoing method transfer over time. Engage QA and regulatory affairs early to understand change control requirements and validation expectations. This is always an interface project. And where internal sources are limited, leverage the expertise of contract laboratories. We have very, very good contract laboratories that have experience with modern endotoxin testing systems. So finally, let's not forget the horseshoe crabs. For decades, they have played a remarkable role in protecting patients around the world. And if equally reliable recombinant technologies are available, then moving towards sustainable alternatives is not only beneficial for supply chain security and business continuity, it is simply the logical next step in the evolution of pyrogen testing. So the goal is not simply to replace one assay with another. The goal is to build a future-proof pyrogen testing strategy, which reduces dependency on aging technologies, strengthens business continuity, improves supply chain resilience, supports sustainability, and ultimately continues to ensure patient safety at the highest possible level. So thank you for listening, and let's push sustainable endotoxin testing forward together as one industry. So if you are planning an implementation project and you are in need of additional input and support, please feel free to contact us down here on the right. You can see our email address. We are happy to help with setting up preliminary testing plans, providing visual step-by-step laboratory protocols and SOPs, and even with hands-on on-site support for the implementation and training in the lab. Lastly, I want to thank some people, starting with the Lonza team for supporting this session, especially Ashley Peterson, Derek Schneeweiss, and Danik Santillan, but also Oliver Ladebeck, Thomas Winkler, and Saskia Ile for the good collaboration and the support during the testing period of the study that I just presented. Then Ian Bedridge and Tony Connell for organizing and hosting the session, of course. Thanks, guys. And also I want to thank the QC and microbiome team at PanPharma. It is so much fun to work with you guys. And of course, Katja Weiss, who was the one who got me interested in recombinant endotoxin testing methodologies in the first place. And lastly, Sarah Riemke. who is a laboratory and project engineer at Harvard Consulting and who is specialized in recombinant endotoxin testing methods and who also supports on-site if necessary. And now I am very happy to answer any questions if the remaining time permits it. Thank you.
Excellent. Thank you very much, Harman. We have got time for some questions, and some questions have come in. There is probably still time to do another one or two. So if you still have a burning question that you'd like, please submit it via the Swapcard platform. So Harman, the first question is, I look at this question, I think that's got to be fairly straightforward. However, I know far better than to actually say that. Is MAT recommended in vaccine QC testing?
That depends. So for products that have shown to be intrinsically rich in endotoxins, I would say yes. Then this should be definitely considered.
Excellent.
Very simple. Yes, it's straightforward.
I do like a straightforward answer, although I noticed you did say it depends, and it wouldn't be a question session in pharma if there wasn't at least one it depends in there. So glad we've got that one out of the way. The second question: what are some of the products you recommend for? I assume for FFC, RCR, and MAT.
RFC, RCR, and MAT. So I have not tested all of them, obviously. I think for RFC, we have had obviously very, very good experience with Lonza. We've also done some testing with bioMérieux, which was almost just the same results, basically. Then for RCR, Charles River, we've had some tests with the customer, and they look very good. So we've also done some things there. And for MAT, I am not very experienced. I've seen some results, but maybe then also obviously Lonza is a supplier. There's also Merck. I'm not an expert. We haven't tried too much of MAT, so I can't say too much of the quality of the different suppliers. For RFC, I was really happy with the systems that we established from Lonza, so I can say that much. Yeah.
Excellent. Third question that's come in, which is, we've heard of low adoption of RFC, for vaccine testing in LMIC due to kit availability and instrumentation costs. Do you have experience of overcoming those kind of challenges in LMIC?
I'm not sure. I don't think I can say too much to this question because we haven't done too much in this area. I've heard some experiences, but I'm probably not the right one to ask for this.
That's absolutely fine. Excuse me. Moving on to the next question. When beginning this transition, and I think one of the key things we always find is those transitionary periods are often the hardest. What challenges are most commonly underestimated in practice?
I think pipetting is hard. I think correct, really good pipe. You can learn pipetting in a day and you can master it in years. But if you do not have a kinetic turbidimetric or chromogenic system established and you're starting with RFC, you have to account for training periods. That's very important because yes, you can learn the assay easily, but you have to be very precise when pipetting it, right? So this has to be trained and practiced. And then I would definitely make a list and see what are my most important products that I want to test. What system is most suitable for this? Can I maybe have a test system and check out if they are working fine on this test system, and then go for the purchase order afterwards and start the project?
I think that's one of the things that people often forget about any new process is that the training is incredibly important. And my experience around that is that often we don't allow enough time to get that training done to the level that we want it to be. Do you think that have you seen that at all, or is that something that is more rare?
No, we actually see this, and you can see this with the CVs also, right? If a new analyst starts using this assay, he will start with very high CV values, and they will gradually go down with experience. So this is something that we see when implementing these systems. And this is the same for LAL as it is for RFC.
How important is flexibility in instrumentation and methods during that transition phase? And following on from that, how should labs think about that when they're making those purchasing decisions?
So I think that depends completely on your business environment also. So are you coming from a very big company and you have a budget that covers all aspects and your only goal is to ensure business continuity? Well then, you purchase multiple reader systems, you qualify multiple suppliers. That is not a problem for you. But this is not the reality in many smaller companies or sites. And there you have, for example, smaller laboratories. You have maybe only one reader system, and you need backup, maybe a contract laboratory, or in case of a reader system that has multiple functions, you can maybe put your old analytical system onto the new reader and still start transferring to RFC. And this is this big benefit that I've talked about the Nebula multimode reader. So you can implement an Nebula multimode reader. You can still use it for all of your LAL-based testing and then start transferring over time. Because if we're real, you have hundreds of products or APIs and raw materials to test maybe. This is going to take a while if your resources are limited to transfer all of these verification studies. To do maybe a 5-year plan, maybe even longer, depending on your business environment. So I think it's really important to have this flexibility.
So I think following on from that a little bit, got a question that says, we are a small QC microbiology laboratory with around 8 employees, and considering how to approach implementation, what would your advice be for those guys? They clearly don't have unlimited budget.
Not only maybe unlimited budget, but it's limited personnel capacity. So depending on your sample numbers and what type of methods you have in-house, you may be completely capped with routine work. And in that case, it's almost impossible to, aside from your routine processes, implement a new system. That's an entire project. It's a different world sometimes. So in these cases, you need to find support for this. Maybe make the list with the most important products. Just write down whatever makes up 50–80% of your testing volume, see the most important ones, and then start maybe checking out on what reader system you can test these. And if you don't have the capacity, do it with a contract laboratory just to start. Then comes the invest, then comes the implementation. And if it's not possible to do it aside from the routine processes in the lab, then find someone who comes in external and helps with the implementation.
I think that's great advice, because this is something that I see not just in pharma but across industries, is that if you are a small business trying to adopt a new process, you're often trying to kind of keep everything running, on clockwork, because you can't stop, and also adopt the new process at the same time, because I know it can be really, really tricky. Another question, you mentioned pipetting, and this is specifically about that. So would automated pipetting bots reduce the errors observed?
So I've had a customer who had implemented a pipetting robot, and there were actually less pipetting errors than when analysts did it. It happened, I'm sure there's different producers, obviously, and maybe there's also different results. But in this specific case, this was true, yes. And I think in the past, I've seen different types of pipetting errors, right? So you can have like a little bit of a splash when pipetting positive controls. It just happens. You don't even see it. It's just so small, and yet still you can get it. This is why we use duplicates when testing. This is why we have this CV value to maybe eliminate one sample if the duplicates are not in line. And this is why we have all of these controls, right? But this happens, and specifically when you have samples very close to the standard curve, to the high concentration standards, this is not a common occurrence, but it's a known occurrence that can happen. Not very often, but it can happen.
So we've got time for, I think, probably one more question. And this one is quite an interesting one, I think. What is the typical timeline from the decision to purchase a recombinant testing system to installation and qualification of the reader? So I think that also plays into the point you made around training as well, because obviously you need to do these things in parallel.
Yeah. No promises. But this is also obviously depending on very many factors. This depends on your own laboratory environment, on obviously supplier that you choose. So when we implement readers, we've had delivery times between 3 months and 6 months. Then you have a qualification period that can be rather brief, but then you want to qualify or maybe do verification studies for the most important products. Then you have some training. So anything between, if you have the decision made, the budget is there, and you say, okay, go, I want the system. Anything between 6 and 10 months, definitely, or maybe 12 months if it's a long period. But that depends on the supplier that you choose, I think.
Excellent. I think that's a good point to close this session. So, Harman, thank you very much, both for the presentation and for staying with us on those questions. What kind of comes through clearly to me is that this isn't a simple drop-in method swap. It's a project, and if you want to do it, there are lots of things that you need to navigate and consider along the way. And if you go in with it with your eyes open, then you will be far better off in that transition process. So our next session is at 3 p.m. That's our third and final session of the day on optimizing contamination control. Excuse me. Big thank you again to Harman. Thank you to everybody working behind the scenes and to our audience as well. And we will see you again at 3 p.m. our time. Oh, and thank you very much to our sponsor for this session, Lonza, without whom we would not be able to put these events on. So thanks again, guys. With that, I will leave you, and we shall hopefully see you again at 3 p.m.